of teaching and then assessments. So yeah, it's fun. But yeah, you all right?
Hello.
Hello. So at what time is it now?
It's 9 p.m. here in Australia.
Okay.
Sydney at least.
Not too bad.
Yeah, much better than yeah, I think because we just had daylight savings end 2 weeks ago, I think it was. But somehow that's changed to 2 hours, so that doesn't quite make
sense. Yeah, that's because you go one way and we go the other way.
Okay, you've had it as well. That makes sense. Okay. I'm just annoyed that now it's dark, you know, when I go home now.
Yeah, the opposite has just happened
now light.
Yeah, you guys are all cheerful.
Yeah.
I always find it funny. I always see like recipes, like, I don't know, websites, whatever else. They're always like, oh, here are some spring recipes to get you, excited for the nice weather. And obviously they are completely wrong for us.
Yeah, the opposite way. Yeah. No, it makes such a difference going home in the lights. Like it's crazy.
It really does.
So, yeah.
So you should send more recipes from Australia then.
Yes,
when it gets balanced.
I suppose so. It's always funny as well trying to remember which things are in season, because that doesn't perfectly translate oppositely, Obviously we grow different things.
Yeah. No, one of my colleagues has literally last week moved back to Australia, so she's sending me all these photos of warm weather. But yeah.
Yeah, I was gonna say it hasn't been that warm, but I guess compared to the same time of year, it's really quite nice. It was 26 degrees today.
Oh wow, I think we're about 6.
But here it's also extremely cold for the time of the year.
Yeah, I mean, I feel like everywhere is having unprecedented weather. Oh wow, really?
Yeah.
So I see that most people are coming in. So I'll give it 1 more minute and then we will start.
Looking good.
So Clementine, you will take the minutes.
Yep.
And I will follow the schedule. Let's see.
Is it me chairing? Oh, you're chairing. Great. I'll shut up then.
Well, you are welcome to chair, but it's your month off. You don't have to do anything now. Only listen and contribute content.
That's great. I tweaked the agenda post, but I didn't add anything major. Just tweaked.
Yeah, and I tried to make it smaller as well. So.
Hello, everyone. Hello.
Hello.
Is this working?
Yeah, we can hear you. Can you hear us as well?
Yes. Okay, it's good. Sorry, I just wasn't seeing it. I wasn't seeing the green light up when you speak on Zoom.
Yeah, I can't see it either when I speak myself. So maybe it's because I'm in the online version. Our university doesn't support Zoom. Okay, then due to the sake of time, let's start, because for some people it's very early, for others it's very late. So we have quite a full agenda for today. We have updated part of the spreadsheet already with the newest in vitro results. Marij, who is here, informed me that she also has a lot of MIC values, so we will update that later this week so that you have the latest values. I noted I forgot to update the in vivo results. So they were updated, and according to the new schedule, I highlighted them in yellow. So if you want to have a quick look on the data which is new, those are highlighted in yellow. We will remove the yellow marker in a month or so, otherwise the whole table will be yellow at some point.
I think we should remove the yellow marker when the compounds have been put into some kind of diagram, right? Otherwise, we are going to miss things, I think. So it's important that someone eyeballs them and somehow shows how the structure is related to potency. There is a risk that we're going to end up with a lot of yellow, but I think that's the only way to make sure that we have all the data and we don't miss something important.
Yeah. I know that for series 5, Andrea is taking a look at them. So probably series 5 will be, the yellow will be removed quite quickly then. Ma is not around. She's ill for now, but that job will be taken over by Marij for the time being. When she's back, she will resume her normal tasks. So if you ask something directly to Ma, it might be that it takes a little bit longer to respond. Then I will go series by series. So series 1. So there was some compounds which we should receive, but we didn't hear about that anymore. So I think they will not come anymore. So should we keep this an item or should we remove it?
Sorry, which compound were those?
So EPL BS 121 and EPL BS 143.
Oh, from DNDI.
Yeah.
Yeah, I think we can archive it, right? Because I just want to put the code somewhere. So just so that we can find them if we need them, because they were gaps. But
yeah.
So if those could be put somewhere on the wiki somewhere, and I don't know how you do that, but molecules that were desirable to be tested but have not been, then we can get rid of them, because I think we're not going to get.
Yeah, me too. So
we can put a note saying take to wiki or something. I don't know.
Yeah. Are there people very familiar with GitHub who can make a wiki for that?
I can just pop it on, like if it's just adding what,
Yeah.
2 compounds, 3 compounds?
To the wiki, yeah.
No worries.
Okay, then that's an action for Clementine. Then we have the paper. So the most recent file is from March 26, edited last at the 23rd of April. Matt, did you take a look at the manuscript?
Yeah, I took a look at it, and the section that Hung has added in on the mechanism of action is really good. So I took a look at the references there, and I just want to have one closer read of it, and then I will sign it off, basically, because I think it's essentially ready to go. And the SAR diagrams are looking great too. So I think essentially the paper is ready to go. And I will clear off the last comments that we had that were some points for you and Ma to look at, which I think you've done all of them. I just want to make sure and then get rid of the comments. So yeah, it's end game now. On me, but I'll do that.
Yeah, so do you want all of the co-authors to have a read after you finalize just before we submit?
Yeah, I think so. Yeah, so I'll alert people when I've had that last read of the final section, and then we can have a last check to make sure everyone's happy. And that's the time when we just need to make sure that we have acknowledged and thanked anybody that we want to thank who isn't an author.
Yeah. Okay. what was the journal where we were going to submit?
I forget, actually. Did we decide?
It was a chemical journal, so I'm not that familiar with those. So
right, don't know. Yeah, I'm not
maybe
sure.
maybe it's on one of the comments. Usually, on my own papers, I make a comment at the title where it was supposed to go to.
Right. I mean, yeah, ACS mention lab or plus NTD would be fine.
Yeah, what about ACS infectious disease?
Yeah, that's possible. Yeah, I think we have a transformative agreement with them, which would cover the open access fee, I think. So we could do that.
Okay, yeah. So there was another comment on the Fennery moles that MycetOS 510 and MycetOS 518 were hit. So do any of the recently tested compounds need to be incorporated? I assume that you will take this into consideration when you read the manuscript, but it's not selected for in vivo. So in vitro, yes, we can add it, but in vivo, it's not prioritized for this season. Also, now Ma is ill, it becomes even more difficult to screen a lot of compounds.
Yeah, okay. I mean, I'm not looking for more compounds to add in. I was just looking for any hits. But yeah, you're right. I mean, if something comes along that's a really good molecule, then that creates an awkwardness for us, right? Because we would have taken that onto in vivo, and we're not going to. So not a big priority. I'll glance at the master list again before when we're finishing off. I'll glance at the master list to make sure that there are no compounds in yellow in series 1 that are, obviously of interest. But I don't think we'll include any more.
Okay, so is there any update on the modeling? Let's see if I don't think Masurian is here. Cyprian is here.
Yeah, and Walid's not, right?
No, so though there are no updates then, I guess, to report. So then we go to series 2, the aminothiazoles. So everything which we have screened is in the master list except of the MIC50s, but those will be updated by the end of the week. There is some new synthesis in progress. So maybe University of Sydney, do you have an update on that?
Yeah, so we have selected some targets for our workshops that we'll be holding in June. I don't have a nice picture of them, but I'll put one up tomorrow or sometime later this week. But we're basically going to try some dichloro compounds that we talked about at the schools meeting and a couple of other things that just explore, adding some rings, etc. Hopefully we'll have about, combining those with some ones that I'm planning to make myself, we'll hopefully have about 20 compounds in the next couple of months, but it'll be a few months.
Okay, that sounds good. Any updates of the SAR? So the structural activity relationship, is that also put into a scheme so that it's easy to analyze later on?
As far as I'm aware, everything was up to date. If you've got a bit of data that's been added in the past 2 weeks, I haven't looked at that yet. But again, I'll have a look at it probably later this week, especially if you've got MIC50s for us there.
Yeah,
but I think
will add by the end of the week. So
yeah, we'll check back. I think it's mostly pretty up to date, and we unfortunately are waiting for some of those compounds that we'll be making later.
Yeah. Any update from any of the others, the schools participating? Does anyone want to give an update there?
I don't know of anything, but Whitgift have made one more compound in this series, and they're going to come to UCL later to characterize it. And the ones that they sent before, I can't remember if they were evaluated. I think they were evaluated. And we have other samples we just need to characterize. But if they're the same molecule, we're not going to send them for evaluation again. Just got to make sure that we've got numbers for them. But yeah, they're pursuing it. And the only thing I was going to raise on this section was the thing about mechanism of action, that if we were going to publish something on this series, we just need someone to do the kind of lit survey of what it is that these molecules are likely doing based on what we know about them. So at that point, you know, I was just thinking if there was a student project, anyone wanted to give somebody to do some lit work, but I don't have a solution for that.
Sorry, I'm just going to jump in for 1 second and say I'm a little bit confused with Whitgift. I think it's Whitgift is the right one, because Desiree from Whitgift has been talking to me about what targets to make, but obviously she is in England and communicating with UCL. So I believe she's got one target left that they are ready to go ahead and make. But after that, I think it might be simpler if Erica is happy to, you know, communicate further from there, rather than have all three of us.
I agree. Yeah, I mean, I think that was the point of the school's meeting, is try to get everyone together. There is some possibility that they may want next time, so next year, to go for series 3, depending on the chemistry.
Yeah, I think we discussed that with Erica, and obviously she's not here today, but I'm sure, you know, if we talk to her about it later, we can get things sorted.
So we also have a batch of aminothiazoles that I'm just going through the characterization to check that they're happy, and then we'll send them off. They're already in on one of the issues. I can't remember which number, but there's the list of the structures there. So we will send those alongside some series 3 stuff when they come through.
Okay. Yeah, with respect to a student doing a literature review, we can look at mode of action, but we are not chemists, so we need a little bit guidance to look to which kind of molecules. There will be starting new PhD student somewhere in the summer in our group, so I can give that student the job to do a literature search, but I need to know what the molecule looks like, so and where to start the search. So I do need a little bit help from one of the chemists to point me in the direction what looks similar, what doesn't look similar.
It would be good to assemble this on the wiki so that we can see it. You know, that would be a good way of sharing the information, and you know, sharing papers with structures where we could look and check that they're similar. So on the, you know, I mean, on the series 2 wiki, we can make sure there's a page there and have a couple of students going for it.
Okay, that sounds good. Any other updates on series 2? Then if not, we can go to series 3. So some of the new compounds, or new compounds in Dutch, showed some promising activity. Yeah, so maybe some updates on series 3, Peter?
Yep, so we obviously sent a batch earlier in the year that we made, and we've subsequently made, I think we've got another 20 to send across. Those students are all finished and writing up now, so it'll be a while before we're back in the lab, but we put some general procedures on the wiki, and I'm currently gathering together the students' online notebooks. Some are in better shape than others, so some need some work. But yes, they're all good. Chemistry is really simple, really quite easy to get a lot of compounds through. So yeah.
Have we gotten any potency data back on them, Wendy?
So for in vitro, yes. And Marij, you did some first toxicity experiment last week. So do you want to share what you found, how they performed?
Can you hear me?
Yes, we can.
Oh, that's nice. Okay. So it looks like after 1 day, yeah, 4 compounds were really nice because from the 15 only 1 or 2 died. So I think that's really nice. 1 compound was actually a bit, I don't know how to say it, but there was a little less survival from that. So
Yeah,
I
so
think,
1 compound showed a little bit more toxicity.
so I think
This,
the 4 are really nice.
yeah, so we will repeat this toxicity test 1 more time, and we will try to do the first efficacy test actually upcoming week. So normally we repeat those at least 3 times, but the first in vivo results will then be generated with the series 3 compounds.
Was those the samples from NEU or the samples from Peter's lab?
Marij, the exact, do you have the exact Myers codes? Which ones we were testing?
It was 631, 632, 628, 627, and 626.
Okay. Let me look it up.
So that's from the NEU compound, probably.
That's great that they're going forward, though, for the next stage of evaluation, so we can get a sense of whether we want to carry on with the series.
Have we gotten any screens from the compounds from Peter's lab yet? Any in vitro? Any in vitro?
I think we're quite up to date with the first screenings. From everything we received was tested in vitro, and only the MIC50 data needs to be uploaded, so the MICs themselves, but all the IC50s are there. From the compounds we received, so.
Okay, I will check that.
Okay, other questions on series 3? Then series 5. We had good potencies for the benzoxazoles. Ma made a scheme, and that was shared to Andrea Ortigo, who's now currently making the structural activity relationship analysis. Apparently, a few of the molecules were missed in the initial scheme of Ma, so I updated that. One of the compounds looked very promising, and we will include it in the toxicity screen as well. So to see if this one can be tested in vivo as well. Ma is starting to write the first draft on the results we found for a paper there. So that's well on its way. There were 2 very good hits which prolonged larval survival. I don't think there's much more updates on that. It's ongoing. Any questions on series 5? No? Then series 6. Dimitri,
Yes.
do you want to share the updates on series 6?
Yeah, I just wanted to say, coming back to our conversation that we had earlier, Wendy, with you this week about the missing in vivo data. So that was something that was missing in the spreadsheet. Now it's updated and highlighted in yellow. So we have all the in vitros now for the series 6 compounds, but we have only 4 in vivo tests for the very first batch of phenarmols that were sent at the beginning of the series when I started them. The problem is that they have been tested in vivo as a mixture of isomers. And out of the entire batch that I have like up to date by now, so this is more than how many, like around like 60, more than 65 compounds if we count all the EZ separated isomers. They have similar IC50, IC90 values. Because they didn't go to the in vivo stage, it's very hard to finalize, get the conclusions on this series. So this brings me to the question, would it still be possible for you and Ma to test at least some of the selected compounds as separate isomers in vivo in the new larvae season?
That
Maybe
really,
like maybe at least 4 or 5 extra. I don't know, because it's very hard. Otherwise to rank them.
Yeah, the problem there are two problems. First, Marij will finish her internship within, well, 5-6 weeks. So I can't ask her to add it to her list. And Ma is currently ill. So that means that her larvae work isn't ongoing yet. So that depends on when she's returned, if we can add them, yes or no. But there's still 20 compounds on her list anyway. So adding a few more, these are not simple experiments which you can do overnight. It takes 3-4 weeks for one run, and you usually repeat them twice. So I can't promise.
Was the reason why you decided to stop the in vivo testing for those compounds? Because most of the catechozymes were very potent in vitro, so based on our gradation system and how we process them to the in vivo stage, they kind of like passed. But then, of course, there are a lot of them. So the problem is there are 2 isomers usually always in those series. We don't have for the project, we don't have them tested separately in vivo, at least not a single entry, which makes it a bit difficult to come with conclusions. Of course, like for those 4 compounds, we have the ratios, and we can assume that like if the E is more prevalent, then it's probably the E form is causing the survival. By the way, the survival rates were not that bad in comparison to all the in vivos that we have for the project. One of the compounds gave 12% survival rate.
Yeah, but that's a little bit difficult to compare. Depends on what the control was. That's also why I make corrected values of the AUC as well. So if in that particular experiment, the control also has a little bit higher survival percentage compared to normal, then the difference between the compound and the control line is not statistically significant. That's also why in the Fennery Mole paper you see different control lines. So we keep the control line with the compound. Otherwise, it's not fair to do statistical analysis, because there can always be biological variation between the larvae batch, between the inoculum. So the survival percentage itself doesn't mean that much when you can't compare it to the control.
Or say, for example, just wanted to add also, say, for example, oxyconazole nitrate, which was tested. So far, it's the best scoring compound in the catechozyme series. It gave like 0.03 micromolar inhibition concentration. Without in vivo, it's very hard. Well, we have this number, but it's also very important in the context that it's a different heterocyclic ring. It's imidazole, I think, if I'm not mistaken. And also it's a salt formulation that also brings us to something that was on development potentially for the future work, as whether we should pursue like salt formulations. And that's why I'm asking, like, maybe if it's possible to do like at least to select like maybe 4 or 5 best compounds in our opinion, and then proceed with the in vivo. Oxyconazole looks very interesting in that.
You could make a listing for us, but still I can't promise. If I don't have the hands in the lab, I simply can't do all the work. It's the same for you. If you have to make 50 molecules in a month, you will probably not be able to do it.
Sure.
and that's for us too. There's a limit in how much we can do. And in the last meeting, we decided to prioritize the ones with a different mode of action above the ones with a similar mode of actions of the azels, simply because all the in vivo data performed so far indicated, also looking at the recent clinical trial which came out, that they will not solve the problem. So even the newest azels, which were much more potent than itraconazole, which is currently used, still could not resolve the infection. Still, you would see, even in the clinic, in the patient, the same phenomenon. And that made us worried that if we still target the same enzyme, we might find the best molecule, which works very well in vitro, which did ravuconazole as well, the study drug from the clinical trial, but it still would not result in vivo, in the real life, in the patient, in a better outcome. And that's why we thought that looking into novel mode of action would be more promising and more relevant. And since we had to make a choice, that was the choice we discussed last time, and we ended up with that conclusion for now. I know that to finalize things, it would be great if we could do all the molecules which were very good at the moment, to finalize and to exclude or to include certain series. But I simply don't have the hands in the lab to be able to do it all.
No, that's understandable. Then in that context, I think the best we can do is just say for the Series 6 is to wrap up this in the general scheme, which is mostly done in my case. So I will update the issue GitHub by the next meeting with the final.
Yeah, and you could make a priority list or a wish list if there was some time over this year. Which one should we go first? That's of course always an option, but I can't promise.
Okay.
But
fine.
if we do have time over, it would be good for us to know which molecules should be tested, have a higher priority compared to others in that series. Is that a solution, maybe?
Yeah. Then we'll just do ranking on the IC90, IC50, and the in vivo for those 4 compounds, so we can still extract some data based on the ratios and other stuff.
Yeah, or maybe
But
also
because it
based
was missing
on
until recently, so the in vivo we just updated it this week. I will come up with the finalized scheme later on.
Yeah, okay, that sounds good. I see that there's some comment in the chat. Oh, it was from Peter that he needed to
Quick
teach.
thing about Series
Other
3,
thoughts?
did the compounds from Peter get tested in vitro yet? Because I did not see the data there.
Do you have a Myers code number? Then
okay,
I can double check.
That would be one moment, just trying to get it. There we go. Okay, let me pull this up. That would be the block starting from. Just going computer load. That would be the block of molecules starting from. That would be the block of molecules starting from like MyOS 658, 657, 659.
Marij, do you know by head if you tested?
No, I have to check it. But I can check it now. I think I will have it in a minute.
Okay.
Yeah, because I just noticed that there was no data published, nothing in yellow, no data So it was, yeah, 659,
No.
6. Yeah. 657 down.
Yeah, Marij is checking.
Okay, no problem. Thank you.
Okay, where did I leave the agenda?
I screened these compounds, but only the screening by 100 micromolar and 25 micromolar because they did not inhibit the fungus at all.
Okay. So did you get that, Anthony?
Yeah.
So the compounds were screened. We will check the list when we are updating the MIC50 that we also upload these data.
Yep. Okay.
Okay. Then we have the global priority box. So that paper was written. And submitted. So I don't think there's much more updates here. Matt, you were checking with GlaxoSmithKline about the availability of more or analogs.
Yep, I wrote and I'm waiting for a reply.
Okay, yeah. And the kinase inhibitor library, let's see if Mickey is here. Yeah, Mickey, do you have an update to share?
I do not really have an update to share, I believe, since last time, because I'm now trying to finalize the manuscript that has been written so far.
Yeah, and we tested one of the compounds from Bernard for toxicity. Marij, could you update us on that?
Sorry, I didn't hear it right.
So the compound, the toxic compound.
Oh, yes, it was
So
the 503. Yeah, all larvae I injected with the compounds, they died. So I will test tomorrow in the lower concentrations if they survive the low concentration, but we will see, I think, in the next meeting if they survived it.
yeah, so I think it was called staurine. Staurine,
I believe.
yeah. Yeah, so that one was very toxic to the larvae.
Yeah,
It was a potent hit.
That one
So
in general, stratospore is pretty toxic.
yeah, so it looks like we will not be able to screen that in vivo, but we will see if we lower the concentration, if the toxicity goes down. But if
And
not, then
the turfostine compound is also tested.
no, we only tested that compound from the kinase inhibitors in vivo.
Yes, there's a turfostine A9 compound, which is also a kinase inhibitor, which was quite active in vitro.
Yeah, so yeah, well, again, we had to prioritize some above the others, but yeah, the first one apparently was very toxic. So other questions about the kinases? Then the plant extracts, well, we never heard back from the plant extracts from Nigeria, so
yes, I wrote
no.
an email, but haven't heard anything from them either. So yeah, I tried again, maybe they were on vacation during Easter or something like that. I will contact them again and maybe they answer and send it again.
Okay. Then the DMPK measurements. Matt, do you had an update on that?
Yeah, so some of the compounds that we sent weren't screened, it turns out, and we got a spreadsheet back from Dundee, the team that did the analysis last year, which explains why. And so I sent that to Dimitri just earlier, just to have a look for the data and assemble a diagram that shows the compounds that were evaluated and what the numbers are, and then the structures of the compounds which were evaluated, just so we can keep them on the radar. And then we can figure out what the data are telling us about how well the compounds are performing, which will be useful to know. I think there's some data in the literature on these compounds, but it's great we've got them measured in the same assay so we can compare.
Okay. Yeah, so I've been in contact with Melissa. It appears that one of her students is actually probably traveling to the Netherlands for another lab here in the neighborhood. So it could be that she will, instead of me sending the compounds to them, she will actually do it in our lab, that student. So I'm in contact with her. That makes life easier with shipping. So to be continued. And I think the toxicity column people did find. I still need to add the data on how we exactly do this, but for now it's simply by injecting the compound in the larvae and watching the survival. If they all die, then it's toxic. If they all survive, then it's not toxic. And if more than 80% is alive and there's no statistical difference between injecting water or PBS and the compound, then they are also still not considered toxic. Of course, there can always be a little bit toxicity, but then it's not toxic enough to exclude the molecule. So we made some rules for that. But I will update that on a wiki. So other things we would like to discuss.
Yeah, I just wanted to say that I started writing a paper about the niclosamide derivatives, which includes mass in vitro and in vivo activity experiments and data. And I think I will send you later this week a first draft of it. It will also include the DMPK data from Dundee, and yeah, we can check then and if there's something else to be done, but I don't think there needs something else to be done from Mars side, because I think it's quite ready for publication as far as I see.
Okay, then I will take a look at it.
Sorry, just one more thing. It feels like it would be helpful for these meetings to have a pharmaceutical industry champion on board, like we have in lots of the other open projects that we are involved in. Someone who comes along and advises from the private sector. We used to have quite a few inputs from Chris Swain, but I think he's just busy with lots of different things right now. So it would be good to try and find someone. I will be reaching out to a few people anyway about this and other projects to see if people can spare, an hour a month to come along to the meetings and give us some guidance and feedback on what we're doing, particularly when we're talking about new targets. But if anybody else happens to have a contact from, currently in or formerly in the pharmaceutical industry who's interested in essentially phenotypic drug discovery, so not target-based stuff, who might be interested in coming along every month, that would be really helpful.
No, the only contact I used to have was in ISI, so, but they were only interested in their own compound.
Yeah, I think that was part of the DNDi network. Yeah, there are also more companies from India, for example, which take part in the DNDi network for preparation of starting compounds or target compounds against leishmaniasis. And yeah, maybe that might be an opportunity. I think Lucia is not here today, but maybe we can ask her about such a possibility.
Yeah, that's a good idea, and it's just very important to try and match the interests of the person with the project. So, there are some projects we run which are sort of fragment projects, and others that are early stage hit identification projects, and you got to make sure that the person that's coming is interested in what you're doing. Otherwise, they won't find it good. But if we can find someone with a bit of expertise in this area of tropical diseases, by phenotypic, infectious, that kind of thing, it will be.
Yeah, for instance, that Indian company, is also preparing compounds which are similar to the Series 2 thiazole derivatives. So maybe that might be a good starting point to include them maybe in Series 2 project.
Yeah, it's a couple of hours a month essentially, right? So it's this meeting and a little bit of prep and maybe a little bit of follow-up, but it's not a lot.
Yeah, maybe they also have access to more libraries or something like that.
I have a question about the niclosamide research. Has that actually been tried in people for mycetoma yet?
Yes, in vivo experiments were done. Toxicity and activity. But the activity results were not significant, but you could see a slight effect that it improves slightly the survival of larvae by some 10 or 15% increase.
Okay, so I was just wondering if there was any cases of it being used off-label to treat people as part of a combination of other things.
No, there's an initiative currently ongoing from Cure ID, where case studies are collected, where people use different medications, also off-label use of drugs, because in some of the countries they simply don't have access to itraconazole, so they use something else. And since there are not so many patients and most of the evidence is anecdotal, they started to collect all the case series, so that if we have enough case series, maybe we can draw some conclusions from that. But that's done via Cure ID in the United States, I think. So, but it will be interesting to see what case studies will arrive from that survey. Any other things?
Just to add to niclosamide, and there are some clinical trials for cancer patients and diabetes patients currently running. Some are already finished. That's what I just want to add, that there are some trials in these fields, but I don't know if there are trials in the field of antifungal research, antifungal infection, fungal infections, sorry. But well, we will see.
Okay. Then if there are no other questions or updates, then I think we can close the meeting. So if you do have a remark, please shout out.
Just
And
about
if
the
not,
recording for this, who owns the recording and who can post to YouTube? Is that the Sydney group of the recording?
I'm assuming it is us. I don't know. It says it's recording. I didn't click recording.
yeah, when you came online, it started recording.
Yeah, okay. Normally I've had to click it before, but I guess it's just doing it automatically now. I was a little confused because I have a recording from whatever the last, well, a meeting a couple of meetings ago. But I was a little unsure where I meant to put it or how I meant to upload it. So do we just put it on YouTube or what?
We do, yeah. So I just did the one for last month. And I can just forward that to you so you've God, actually, in the issue for last month on the 26th of March, there's a link there now to the YouTube recording if you wanted to check that out. And essentially, I've just put it from myself and just added a bunch of hashtags and keywords so that it can be found. We don't really have a more advanced solution than that.
Okay, that's fine. I just I remember I went to upload it at some point and just wasn't sure if there was a specific thing I was meant to be doing.
No, not really. No, we toyed with the idea of a shared account, but it's just too difficult to administer.
Yeah, fair. All right, I'll try and upload both of them soon.
All right, cool. Thanks.
Okay, then the next meeting will be on the 28th of May. And remember, it will be on the alternative time. So that will be early morning in Europe and Africa. So it will be a few hours earlier for Clementine as well. Yeah, it will be for everyone a few hours earlier, I guess. Okay, then see you all next time.
Great.
Bye,
Thanks all. Bye.
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 Wendy, do you have 2 seconds to talk? Sorry.
Yes, of course. Do you need to stop the recording for that?
Yes, that's a good idea. I'll